Hi students, welcome back. Last time we left off on the anatomy of the ear, and I mentioned that it was really important to understand so that we could kind of understand how hearing works. And not only hearing, but how we sense gravity, like where we are kind of in relationship to the surface of the earth, right? So are we falling left, are we falling right, are we moving forward, are we moving backwards, going up in an elevator, and or going down? That's what we mean by gravity. But also linear acceleration. So are we moving forward, walking forward, not leaning forward, but walking forward or walking backwards or walking to the right, etc. So we're going to go over those types of sensations, and we're going to focus on what's called the vestibulocochlear complex. That is this entire structure right here. And we're going to look at probably something you've already heard about before, the semicircular canals, the vestibule, and then also a separate structure called the cochlea. So let's go ahead and I'm going to erase this real quick. And the first thing we're going to learn about is how we process sound waves, or how those sound waves are taken from the external ear through the middle ear and into the inner ear. So if we go back, I'll use a green. If we look up in the upper left-hand corner here, let's just kind of imagine sound waves, whatever it is you hear, my voice, your dog barking, a child crying, you name it. Those sound waves are being collected by the external ear. That big external ear you see there acts like kind of like an amplifier, or sorry, not an amplifier, a structure that collects sound. And then those sound waves enter the external acoustic meatus, and the external acoustic meatus amplifies the sound. So we've got sound waves traveling in the external acoustic meatus, and eventually what's going to happen is those sound waves are going to bang on that tympanic membrane. And that tympanic membrane is connected to the malleus, which is connected to the incus, and which is connected to the stapes. So up until this point, the ear is just collecting sound waves and transmitting those sound waves to this vestibulocochlear complex. We haven't actually physically heard anything yet, or perceived any sound in the brain. All of that magic occurs afterwards, the perception of sound and the interpretation of sound. But we need to get these sound waves that travel in the air into this structure somehow, into the cochlea. And the reason for that is the cochlea has a specialized structure called the organ of Corti, which has sensory cells in it. And those sensory cells are going to be stimulated by sound waves. And then the sensory cells then are going to send an action potential along the vestibulocochlear nerve, or the auditory nerve, back to the temporal lobe of the brain for initial processing. So you can see kind of the conundrum that we have here is that we've got sound waves traveling in the air, but sound waves themselves aren't going to trigger the brain to perceive much about sound. We've got to transform those sound waves into something that is electrical activity. You know that neurons work on electrical activity, and those sensory receptors, those neurons, are located here all the way up to the top of this cochlea right here. So we're going to have to transmit that or change this form of energy in the energy of sound waves. We've got to change that to electrical energy. And so let's come back here. All right. So I'm going to, again, erase this. And let's zoom in here. So like I said before, let's imagine sound waves. They're going to be collected by the external ear, and then they're going to travel within this external acoustic meatus. They are going to tap or cause moving on the tympanic membrane, which is this structure. And the tympanic membrane, as you know, is connected, like I said, to the malleus, which is then connected to the incus, and which is then connected to the stapes. Well, great. Now we've got actual bones moving, right? Those auditory ossicles, those are moving. Before, we had energy just traveling within this external acoustic meatus, and although molecules in the air were moving, that's not really something you can see, but now we've got this physical movement of the malleus, the incus, and the stapes. And the stapes is connected to this vestibulocochlear complex at a structure that I'll show you right here. Oh, it's not on this picture. It would be right about here, called the oval window. So let's imagine that the stapes is right here, and imagine that stapes being pushed by the sound waves, right? All those auditory ossicles are moving. So it's going to push on this oval, that oval window right there. And this entire complex, this entire vestibulocochlear complex is filled with fluid. So when that stapes part starts pushing on this oval window, and that's just a little membrane, that causes the fluid inside the vestibulocochlear complex to start vibrating, to start moving around. Imagine taking your arm and sweeping it through the surface of the water in your bathtub or a swimming pool. You physically see the energy that's been transferred from the movement in your arm as waves on the surface of your swimming pool or on the water in your bathtub. Really kind of something very similar happens here. And so let me go back and erase this. This is just such a cool process. I get really excited about it. Okay, hold on. I gotta erase some more here. Okay, so you're like, Bridget, well, this whole structure, this whole Cinnabon-looking structure, this whole cochlea, yeah, it's filled with fluid. That's great, but I still don't understand. Well, let's look at the three tunnels, or the three ducts, like this, not like duck as in the flying bird, right? Let's look at the ducts. So within this vestibulocochlear complex, there are three separate passageways or ducts. The first one is what we call the vestibular duct. That's this one on top, right here. And that duct is going to go all the way up. You're looking down on this snail or down on this Cinnabon, so this would be that passageway. Okay, so that vestibular duct is filled with a fluid called endolymph. That's the name for the fluid that we find inside the vestibulocochlear complex, inside the whole inner ear, really. Well, there's another duct called the cochlear duct, which is right here. And this blue structure, that is a separate passageway that runs right alongside the vestibular duct. And it's also filled with fluid. And then lastly, at the bottom here, that's what we call the tympanic duct. And that tympanic duct, also filled with endolymph, runs right alongside that cochlear duct. And they maintain their orientation with one another, even though they kind of spiral around one another like a spiral staircase. They don't change their position in relation to one another. They're always right next to one another. So let's think of these as fluid. They're all fluid-filled passageways. But one of the big differences between these three is that the cochlear duct, the yellow duct right here, this one in the middle, that is, imagine the walls of that cochlear duct being made of a very thin, very soft membrane. And let me get my highlighter here. And the vestibular duct and you can see it drawn in brown, this brownish beige color, and the tympanic duct, well, they're filled, they are filled, they are surrounded mostly by bone, whereas that middle, hold on here. Okay, I've regrouped here. Let's first look at the vestibular duct. This vestibular duct, most of its walls that you see here drawn in brown, they're bony. And if you look at the tympanic duct, the one on the bottom, you can see it's surrounded in that beige color. Most of its walls are bony, but they surround this passageway in the center, that cochlear duct. And the cochlear duct, its walls are very soft, very thin membrane. So imagine this cochlear duct also being filled with fluid, and it's like a water balloon between two pieces of PVC pipe, where the PVC pipe has solid hard walls, and that would be like the vestibular duct up here and the cochlear duct down, the tympanic duct down here. Think of those as hard-walled, almost like PVC pipe. And sandwiched between the PVC pipe is a water balloon filled with water. So that's kind of the analogy I want you to think of. Well, remember that we've got let me switch to my pen. We've got the oval window about right here. And then we've got the stapes, that bone, that's going to push on the oval window. So we've got this motion of bone, like this physical movement of bone, now pushing on that oval window. And that oval window is a really thin membrane too. We'll just color that in, just because coloring is fun. So we're just going to color that in. So now we've got energy being transferred to the fluid inside the inner ear. So you got the stapes pushing on that oval window, and that oval window is going to move in this direction, right? So it's like pushing your hand through water, or pushing your hand across the surface of your bathtub. You're going to move some water, right? So you're transferring energy from the movement in your arm to the water itself. And what's going to happen is that movement starts to cause waves or cause movement of the fluid in that first duct, the tympanic duct. But as that movement in that fluid, it'll continue to travel all the way around that cochlea, right? And that depends how far these sound waves move totally depends on the frequency of sound, but we're not going to go into that. So you've essentially got waves being created. Within that the vestibular duct. So you're like, well, great. So what do waves do, right? Waves still don't translate to electrical activity in the brain. So as this fluid begins to move, like this, kind of up and down, because this membrane of the cochlear duct is so thin, some of that energy gets transferred into that cochlear duct, into that soft membranous duct, and starts to move the fluid inside there. So now you start to get fluid moving in that cochlear duct. And what's special about that cochlear duct is, like I mentioned before, this structure called the organ of Corti. So what we're going to do on the next slide is we're going to zoom in on a section of that cochlear duct and look at the organ of Corti. Let's go back and look at, so now we can go back to this picture. And it's not shown very well on here. If I were an author of a textbook, I would have shown it better. But alas, nobody has asked me to author a textbook, so maybe we'll leave it to them. Okay, remember? We got that energy traveling through the vestibular duct into the cochlear duct, and then ultimately into that tympanic duct. What they're not showing you here is another little window, a membranous window, called the round window. Remember there was that oval window where the stapes was pressing on? Well, now we've got another window called the oval window. And so that energy that's in the fluid of the tympanic duct is going to simply just leave right out that cochlear duct. Right out that tympanic duct. So let's move to next slide here. So let's go ahead and do that. So now the way we're looking at this is more of a, it's a section. It's a cross section through the tubes. So you've got the vestibular duct here. Let me use a different color. We've got a vestibular duct. There is the cochlear duct. And then there's the tympanic duct. So you've got movement of fluid. You've got movement of fluid in here, right? So this fluid in that vestibular duct, that's moving around. But some of that energy, as the fluid moves around, is going to pass right through that membrane and start moving the fluid in the cochlear duct. Right here. So now we've got this fluid bouncing around, which is great. That's exactly what we want. The organ of Corti is the structure right here. So let's go ahead and look at that in a larger view. Let's imagine now that we've got fluid moving around in this chamber, and what that fluid is going to do is going to cause this entire structure to move up and down. It's going to kind of bounce around, right? So think of this structure as being like a floaty in your pool. Somebody starts to make waves in the pool, and you're going to float on the surface of the water in your floaty or in your inner tube or whatever it is. So this structure on the bottom begins to move, and this structure is called the organ of Corti. And let's look and see what it's made up of. You've got some supporting cells here, which are drawn in blue, and that's not that different than what we looked at when we were learning about the sense of smell, right? You had those columnar cells that were acting as supporting cells. And what they've kind of done is sandwiched these specialized, what we call hair cells, right here. Now these themselves aren't neurons, but they are specialized cells with stereocilia on their surface, really long cilia on their surface. And at the base, you see that these stereocilia cells, or these hair cells, they're going to synapse. They synapse with a neuron. So down here, these little branches, those are the dendrites of a bipolar neuron. Okay, great. Now you've got a synapse. Now you've got a connection between that cell, that sensory cell, that hair cell, and the neuron. So when this organ of Corti starts bouncing up and down, what's going to happen are these little hair cells here, they're going to come in contact with this pink finger-like structure called the tectorial membrane. And when that happens, these stereocilia, I'm going to use green just to show you what the stereocilia look like when they're bent. When the tectorial membrane comes down and comes in contact with those stereocilia, these stereocilia get bent in such a way that causes this hair cell right here to release some neurotransmitters that stimulate that neuron, essentially sending an action potential back to the brain to be interpreted as a car driving by, your professor's voice, your child crying, whatever it is. This structure right here is a collection of axons, and you know what we call a collection of axons, right? We call it a nerve. That is part of, and you can see it in larger form here. right? That is one branch of the cochlear branch of cranial nerve 8. Remember, the cranial nerve 8 is also called the vestibulocochlear nerve. So there are two parts to that. There's the vestibular branch and there's the cochlear branch. We're looking at the cochlear branch. Can't see it there. Slide, the big picture. So this right here too would be a branch, one of the branches of the cochlear branch of the vestibulocochlear nerve. So we essentially have, if we look up here, we've got energy, and I'm just going to draw it as one line. Energy traveling in the fluid in the vestibular duct down through this cochlear duct, and that causes the basilar membrane or the organocorti to start bouncing up and down. That bouncing up and down causes the hair cells to come in contact with this tectorial membrane, and that then causes an action potential and causes the neurons to fire and send information back to the brain about what's being heard. Now, this is energy. You got to think of these waves as these sound waves as a form of energy, and we can't let energy build up in an enclosed space. It's got to get out somehow. So this is really where the role of the tympanic duct comes into play. This is a one-way movement of energy. So we've got that energy in the cochlear duct in the form of the movement of fluid. Well, that energy just simply continues to move through that cochlear duct and into the tympanic duct. So I'll do it one more time. You've got this simple transfer of energy that's moving through 3 fluid-filled ducts. That begins in the vestibular duct, travels through the membrane of the cochlear duct, through the basilar membrane, and ultimately that energy ends up right there in the tympanic duct. Now you're probably like, well, I don't get it. So that energy ends up in the tympanic duct, which is here, right? This bottom duct, but that energy is still caught up in the vestibulocochlear complex as a whole. Oops, not that picture. This picture right here. We've just kind of tracked that energy traveling down that tympanic duct, and there is that oval window. So that excess energy is going to end up in the middle ear right here, and ultimately then be released down into the oral cavity through this auditory tube. So any energy that builds up, or any energy traveling through that endolymph in the cochlea, we can't let it get trapped in here, right? So it kind of it causes that movement in the fluid, and it's that energy's traveling through the fluid, remember? And it's going from from vestibular duct through the cochlear duct down into the tympanic duct, and then it's going to kind of make a U-turn and end up in that middle ear and be released then into the air in the auditory tube. And the auditory tube leads to what's called the nasopharynx. It's essentially a part or region of the back of your mouth. So that's how that excess energy gets out of the inner ear, so we don't cause damage to the delicate structures inside the inner ear. Okay, so that's the production of an auditory sensation. That's just the anatomical way of saying production of sound. And remember that occurs here in the cochlea. And ultimately what ends up happening is an action potential is sent down this cochlear branch of vestibulocochlear nerve, and then ultimately back to the brain for processing. Now let's focus on the other areas of the vestibulocochlear complex. There is the vestibule, which is this area, and then there are also the semicircular canals, these structures that you probably heard of. So they're responsible for the perception of different sensations. The linear rotational movements, linear acceleration, and also gravity. I'll show you on another. So let's go ahead and look at the semicircular canals. The semicircular canals are responsible for allowing us to perceive rotational movements. So rotational movements might be like spinning around. That's a rotational movement. Falling forward, falling backwards, falling to the right, falling to the left. Your perception of getting up out of bed in the morning. Your perception of laying your head down in the evening or at nap time or whatever it is. So just like the cochlea, the semicircular canals are filled with endolymph. And let's look at that. You don't need to know which semicircular canal is which, but there's anterior, posterior, and what we call lateral. In this blue passageway right here, that's where you find this fluid, this endolymph. And as you move around, you make different types of movements. That endolymph kind of sloshes around back and forth in here. So it's going to go back and forth. And it's going to do that in all three of these semicircular canals. Just kind of depends on the type of movement you're making, right? So it's going to slosh around. At the base of those semicircular canals are highly specialized structures called ampullae. And they kind of, in this picture anyway, look like hourglass figures. And if we go and kind of zoom in on what an ampulla looks like, you're going to see some really familiar things here. You're going to see the fluid. I don't know if you can actually see it, but it's drawn here in blue. So you're going to have the fluid around this little gel cap. Okay, so that's fluid in blue there. And then this little gel cap, this little gel cap right here that you see in purple, that's called the cupula. That's the term there. And that sits over the top, that cupula sits over the top of these hair cells. We've seen hair cells before, right? So here are the hair cells, the cell body itself, and here are the stereocilia. And they're embedded in that cupula, that gel cap, right? So if we've got fluid moving in one direction or another, depends on what type of movement your head is doing, that fluid is going to push on the ampulla. And because those hair cells are embedded in the ampulla, these hair cells right here, the stereocilia, they're going to bend as well. And just like before, when they bend, let's just pretend they're going to bend in this direction. When they get bent in that direction, that causes the hair cells here to release ions that stimulate a nerve, and that ultimately sends information back to the brain about the type of rotational movement. So are you nodding your head? Yes. Are you shaking your head? No. Are you leaning or tilting your head from side to side or tilting your head backwards? We perceive all of those things through these specialized ampullae, this little organ right here, that's found at the base of each one of these semicircular canals. So far, we see a setup or a structure that's really similar to the way in which olfaction or the sense of smell is perceived. You've got supporting cells, and here you've got supporting cells, right? You've got specialized cells that are specialized for being triggered by a specific movement or a specific molecule. And then you've got nerves carrying that information to where it will be processed in the brain. Saw that with olfaction. We saw that with hearing a few slides ago. The same is true here with rotational movements. Now what we're going to move on to are linear acceleration and gravity. So the movement of driving forward or walking forward or driving backwards or walking backwards. So linear movement refers to our movement horizontally across the surface of the earth. And gravity then refers to our position with regards to the surface of the earth or your floor or whatever. Think about it as like the feeling you get going up in an elevator. Are you moving up or are you moving down? Think about the feeling you get on a roller coaster. You perceive that you're going up, 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 up the hill, right? So that's a gravitational sort of perception. And then imagine the feeling that you get when you are coming down the other side of the hill on the roller coaster. That would be moving fast down, back down towards the surface of the earth. In fact, a lot of theme park, amusement park rides play on the anatomy and physiology of these semicircular canals and these structures, these sensory receptors that we find in the inner ear. So let's move on to the structure of what we call the utricle, which is here, and the saccule, which is here. There are specialized sensory structures inside of those two things that are called maculae. So on this picture right here, there's a macula inside of the saccule, and there's a macula inside of the utricle. Let's take a look at a slightly better picture of that. Okay, so we just looked at the saccule and the specialized structure inside called a macula. And there's another one over here, right? And that's another macula inside of this swelling called the utricle. So if we were to zoom in on that, we see that the macula is made up like this. Look at some of the similarities first. You've got supporting cells, which we've seen already, right? There are hair cells that you see here with their stereocilia on the surface. You see those hair cells synapsing with a neuron. And we also see this gel cap. Now it's a little bit longer here. It's more like a layer of frosting of gel, so to speak. Now what's different is that we have free-floating structures that are not attached to the gel, the gelatinous material, and those structures are called statoconia. Those are little crystals. So since they're free-floating, they have the ability to move or slide across the gelatinous material back and forth, or float upwards. For instance, if you were to go down in an elevator, for instance, this part, which is attached to the rest of your body, that's going to fall, right, as you go down in the elevator. But these little statoconia, they're not attached. So for a split second, they put just a little bit less pressure on this gelatinous cap. And that lessened pressure then is perceived by the brain as this falling feeling. Now imagine, for instance, if you were going up in an elevator, we all for a split second feel a little bit heavier. And that's again because this section, this part of the macula right here is attached to your body, but the little statoconia are not. So for a split second, you this part of the macula is pushing up on those statoconia, which then bends those little stereocilia. So that would be how we perceive gravity. But I haven't addressed yet how we perceive linear acceleration. So I'm going to erase all this here real quick. And let's talk about this, linear acceleration. You can use the analogy of driving in a car, running forwards or running backwards. I like the car one, and I'll tell you why in just a second when we get to this. Okay, just a couple of seconds ago, we learned that these little crystals, the statoconia, are free-floating. They're free-floating on top of that gelatinous material. Well, that means they have the ability to slide backwards or slide forwards depending on what the rest of your body is doing. Remember, this area of the macula, that's connected to you, and you are connected to your car, or you are connected to the ground, or whatever it is you're on. So this bottom area of the macula is going to carry out the same movements that you do. Statoconia are going to float freely. So imagine you're sitting at a stop sign, in a car, not at a car, at a stop sign, in a car, and you accelerate. What happens is that these statoconia are going to slide backwards. Think about what happens to your purse or your gym bag or your groceries in the front seat. What happens, and you feel this too, right? What happens is your groceries slide back as you move forwards, right? So that's just like the statoconia. The statoconia are sliding backwards in this direction, and that causes the cilia to get bent. And that causes these cells to generate an action potential, to release ions that cause these nerve cells to generate an action potential that gets sent to the brain, and your brain goes, aha, I'm accelerating linearly, I'm moving forward. Now this happens to me all the time, whether it's my dog in the front seat or my purse, but you're cruising along, and all of a sudden there's a stop sign, and you abruptly stop. This part, let me change colors here. This part, again, of the macula is attached to you, and you're in your seat in your car, and your car's wheels are attached to the road. So everything stops, right? Except for the statoconia. The statoconia continue on it takes a couple of seconds for them to catch up. So those statoconia, they keep sliding forward, even though your body has stopped. So if this is your bag of groceries or your purse, that ends up on the floor. Or your dog, my poor dog did this. Not intentionally, but I had to slam him on the brakes. He was in the front seat. I should know better. But he fell right off the front seat and into the little space between the dashboard and the front seat because he wasn't seatbelted in. Had he been seatbelted in, would have been fine. He wouldn't have moved. So those statoconia, they slide forward, and that bends these cilia, use a different color, bends these cilia in a different direction, which causes this cell to release ions, which then initiates an action potential in this neuron, which then gets sent to the brain, and the brain goes, I've just come to an abrupt stop. So that would be like an example of deceleration. So it's a linear movement, right? But we're not accelerating going forward. We all of a sudden stopped accelerating. You can think of that as deceleration, if you like, slowing down. So that's linear acceleration. So we learned about how these statoconia give us the sense of gravity. Are we moving up? Are we moving down? And how those statoconia allow us to determine if we're moving forwards or backwards. All really important sensory stimuli that the brain requires to maintain homeostasis. Okay, the last half of this special senses lecture really is just made up of pictures of the eye. And since you are working on the cow eye dissection, it would be a little bit redundant for me to read what I have here. I'm not going to ask you about the details of the function of the eye beyond what is written here on these few slides. So really, these slides are showing you the structure of the eye and then telling you basically just the basics of what some of these structures do. And the reason for that is anatomy is anatomy. Yeah, we're learning a lot about structure, not so much about function, which is the physiology. But it's my understanding that physiology instructors will cover this when you get to physiology. Or if you've had physiology, then you understand how the eye works. So you'll just have to know and then anything else that you found in the cow eye. Okay? Thanks for joining me. If you haven't done your dissection yet, get to it. It's a lot of fun. Okay, see you later. The information that's on these slides, and if you did the cow eye dissection, fantastic. And you've seen all listed here. these structures that are listed here. You've seen all of that in your cow eye. I hope that you all is what I really enjoyed that. I know it's a really fun thing for us to do in class together. So for the eye, all you need to know